The next most important topic that we are going to study is about enzymes. First of all, we need to understand that whenever we use this word enzyme, what is N? N means in. When I say zyme, the word zyme basically with respect to yeast, in which there was one enzyme present that is called as zymase. So in this way, the name enzyme came into picture. Enzyme basically means in yeast. So this term that you have studied enzyme, it was given by one of the great scientists in the name of William Cohne. And William Cohne in 1878 was the first scientist ever to give the term enzyme. The point here is who isolated first. The term enzyme was given by William Cohne. The first scientist ever to isolate enzyme was J.B. Sumner. And in the year 1926, J.B. Sumner isolated enzyme. And he isolated urease. Urease was the first enzyme to be isolated, and it was isolated from jack beans. When I say jack beans, it is jackfruit. We all call it as fungus. Let's understand. If there is a cell and the cell is performing various metabolic processes. So if I want the metabolic process to go very fast, I must use enzymes. So enzymes are basically the most important component that helps to increase the speed of the reaction. So speed of the reaction, if I want to be increased, I must use enzyme. Otherwise, the reaction will take place very slowly. And whenever we talk about reactions, we need to understand, students, that reactions takes place at a particular temperature. Reactions will take place at a particular pH. Let's understand what are the various physical environments required for any enzyme to work. So here we are going to focus on very first one, that is called as physical environment. And if this physical environment is available for the plant, for the animal cell, then only the enzymes can work very properly. So the different physical environment. First physical environment is the temperature. There has to be a proper temperature so that the cell's reaction takes place properly and enzymes can work. Second physical environment is the pressure under which the enzyme is working. Then the condition, whether the enzyme requires alkaline condition or the enzymes required acidic condition. Based on the conditions, the enzymes are going to work. And the most important is body's requirement. If body needs that enzyme to work, then only the enzyme will work. So based on these factors, the reaction will be decided. Now the point here is we use a very important and a technical term that is called as enzyme. Enzyme, what are enzymes? If somebody asks you what are enzymes, so what you will say? Just remember, students, enzymes are nothing but these are specialized macromolecules. Enzymes are specialized macromolecules. All the enzymes basically are proteins, but all proteins are not enzymes. And most important is enzymes are biocatalyst. Now you might be thinking, sir, what do you mean by this word biocatalyst? So let me explain. Biocatalyst means, first we need to understand the word that is called as catalyst. Catalyst is that organic compound that will always increase the speed of the reaction but never participates in the reaction. It means what a catalyst? It is that chemical that will always increase the speed of the reaction. It never participates in reaction, and as a result, what happens? it remains unchanged. This is what is enzyme. And when I say biocatalyst means the catalyst that is involved in biological reactions. I hope this is clear. If it is clear, put it in the chat box. Yes, sir, it is clear. Moving towards the next important part, that is called as types of enzyme. Now, this is very important to understand. When I talk about types of enzymes, we need to understand that there are two types. Very first type is endoenzyme, and the second type is exoenzymes. Now the point here is, what do you mean by this word endo and exo? So when I use this word endo, it means in. When I use this word exo, it means out. So what exactly is endo and exo? So remember students, endoenzyme means the site of synthesis of enzyme and the site of action of enzyme is same. That is what we mean by the endoenzyme. For example, if this is the cell and let us take this cell produces the enzyme A. So enzyme A is produced by the cell and this enzyme A is going to act on the same cell. Then this kind of enzyme is basically endoenzyme. When I say exoenzyme, what exactly it means? It means that the site of synthesis and site of action is different. What exactly it means? Here, the enzyme will be produced from one cell. If this is the cell and it is producing certain enzyme, so this cell's enzyme will not act here. It means, let's take B to be the enzyme. So this B enzyme will not act on this cell. It will travel via some source and it will reach on this cell to act. So this B basically is nothing but this cell is the site of synthesis, and this cell on which the enzyme is acting is the site of action. So we can say it is produced from one cell and it is acting on other cells. This is what is called as exoenzyme. Most important statement that you need to keep it in mind is that most enzymes are proteins, but all proteins are not enzymes. If all proteins are not enzymes, then what they are? They can be hormones. All proteins are not enzymes. So this is very important statement that we need to understand. Next part we need to understand is the nature of enzyme. Now when I say nature of enzyme, it means how it is going to act, what is the mechanism of action. These are the two important points that we need to understand. So let's start with nature of enzymes. So when I say nature of enzyme, what exactly it says? So it is purely based on the chemical composition. So I will say based on chemical composition, I can say that the enzymes basically they are categorized into two major parts. Which are the two parts? The very first part is purely proteinaceous enzyme. So when I say purely proteinaceous, it simply means that it is made up of only and only protein. And the second type is conjugated enzyme. Now when I use this word conjugated, we all have studied in proteins. Conjugate proteins. Conjugate proteins means it will have two parts. One part will be the protein part and second one will be the non-protein part. So I can say that the protein and the non-protein part together makes up the conjugate enzyme. And this non-protein part, students, remember, it is called as prosthetic group, and it is known as prosthetic group. So what are the examples that we need to study? In purely proteinaceous enzyme that we can say, there is a protease. Now when I say protease, the word protease means the one that is going to break protein, and the prosthetic group basically is like glycoprotein, you can say, that becomes the conjugate protein. So these are the two different categories. One is normal and second one is bit different. Let's understand about conjugate enzyme. So whenever I say conjugate enzyme, so students, what we need to understand here, whenever I use this word conjugate enzymes, so you have understood that there are two parts involved. First part is the protein part. The second part is the non-protein part. And that non-protein part which we have studied basically is nothing but it is called as prosthetic group. Now this prosthetic group has certain conditions. First is that it is firmly bound to the protein by chemical bond. So non-protein parts is firmly bound to the protein component. Very important line that we need to always remember. It is firmly bound to the protein component by chemical bonds. And remember students, this bonding is very strong. That's why we say, and it is not removed easily by hydrolysis, and it is not removed easily by hydrolysis. Now this becomes the very important statement as far as non-protein part is present. If you might ask a question, sir, if this non-protein part is removed, then what? So remember student, if non-protein part is removed, it means you have removed it by force or by some kind of action. Then what will happen? Then protein part of enzyme, protein part of enzyme becomes inactive. Very important part. Protein part of enzymes becomes inactive. So logically, what exactly it wants to say? Let us take this to be the enzyme, and this enzyme is nothing but it is the protein part, and along with the protein part we have added some non-protein part. So this non-protein part is basically prosthetic group. So let me label it. This is protein part and this is non-protein part, which we all call it as prosthetic group. So it is bound very tightly by the help of chemical bonds, and if it is bounded, then this enzyme will work. So I can say that the enzyme is active, but by some force. What I will do, I will remove this prosthetic group and only enzymes are formed, only protein part is left out. And remember student, if only protein part is left, and what we have done, we have removed basically the prosthetic group. It means it is somewhat like this, protein and the prosthetic group. So the prosthetic group is separate. So what will happen under such condition? Under such condition, I will say now this protein part is not active. So remember, the conjugate protein has a very important role. Now, there are enzymes which require certain organic compounds and inorganic ions for their activity. So let's take the another topic. What I will say that there are enzymes which requires, so there are enzymes basically which requires two things, either one of them. One will be organic compounds. There are enzymes which requires, first I will say organic compounds if required, and even inorganic ions. So this becomes the very important part. There are enzymes which requires organic compounds and inorganic ions for activity. Now you might be wondering, sir, what is that term? What is that compound called as? So if it is the organic compound, remember students, I take this as the protein part. Now I take it this we have taken in the diagram also. So let's take this to be the protein part. Now to this protein part, if I attach organic compound, so let us take this. Now this is nothing but the organic compound. So what it will be called as? So remember, to this organic compound, if protein is tightly attached, or I can say to the protein part of the enzyme, if organic compound is tightly attached, then it is called as coenzyme. It is called as coenzyme. Remember, this is very important word. So the coenzyme is nothing but it is the organic compound, and most important, what we need to remember, that it is tightly bound. It cannot be easily removed. And if I take the same diagram here, where I will be drawing a protein part, and to this protein part, let us attach inorganic ions. The very important part here is that this inorganic ions will not be tightly bound. It will be loosely bound. So let's take this to be the protein. Let's take this to be the iron. Now this iron is basically the inorganic ion. Now this inorganic ion which is attached to the protein is loosely bound, and it is called as cofactors. Very important. It is called as cofactors. And most important point, that they are loosely bound. So with this we understood two important statements, that organic compound is a Bahubali type which is strongly attached to the protein part, and this inorganic ion is not strong, it is loosely bound, and it is called as cofactor. Let's understand some examples. Let's understand some examples. So first we are going to talk about organic coenzymes. Let's understand very first example, that is organic coenzymes. So for this, the example that comes in my mind is NAD and FMN. You might be wondering, sir, what is NAD? What is FMN? So when I say NAD, it stands for nicotinamide adenine dinucleotide. That is NAD. And when I say FMN, it stands for flavin mononucleotide. So flavin mononucleotide becomes the most important. These are organic coenzyme. Let's understand the another example. Now another example that we are going to focus on will be with respect to inorganic ions of metals. Inorganic ions. So when I take this inorganic ions, now these inorganic ions that we are going to talk about, there are certain metals basically which are going to play an important role. So let's understand the different metals. So the metals that can be used as a cofactor, metals which is going to act as cofactor. Now those metals can be basically, I can say magnesium, that is Mg, it can be copper, it can be zinc, it can be iron, it can be manganese, and I put it as etc. Now this becomes the important part. So let's understand for Fe first. So when I use this word Fe, remember students that Fe is a cofactor. Fe is a cofactor for enzyme catalase. Fe is a cofactor for enzyme catalase. Now this catalase is the enzyme and Fe is the cofactor. When we talk about manganese students, so manganese is nothing but it is acting as a cofactor for one of the enzyme that is called as peptidase, the enzyme that is going to break the peptide bonds. So remember, metal cofactors, they are referred to as enzyme activators. Very important statement for your exam. Metal cofactors. Is there any other name given to metal cofactors? The answer is yes. And they are called as enzyme activators. Means they are going to activate the enzyme. And if metals are not available, then that enzyme will not be activated. Moving towards the most important characteristic, that is nothing but the properties of enzyme. We need to focus on properties of enzyme. So when I say properties of enzyme, what are the different properties? I hope till here things are clear, everyone. And when we talk about properties of enzyme, so what we can say, that all the enzymes, they are very much important for any enzymatic reaction to take place. So but the very simplest and the important point, that enzymes, they will always accelerate the rate of reaction. So I can say enzymes accelerates the rate of reaction. Enzymes accelerates the rate of reaction, but most important, they do not initiate it. It's like a fight going on between two-person. If two people are fighting, what will happen? The third person will come and say that he is trying to fight with you. Here he is using abusive words. So the first person again starts saying, why did you told me like this? It means the third person has not started the fight. The fight has been already started. The third person is just acting as an enhancer to just provoke or to increase the speed of the fight. That is the role of the enzyme. It will never initiate the reaction. It will always accelerate the reaction that was started. And most important part, that the enzymes do not participate in the reaction. Enzymes do not participate in the reaction. So enzymes do not participate in the reaction. Or and you can say, as a result, since it is not participating in the reaction, as a result what happens, it remains unchanged. So it remains unchanged at the end of the reaction. It remains unchanged at the end of the reaction. It means there will be no change taking place in the enzyme as well as in the reaction. To understand this, let me put it one very easy and interesting example for you. चलो ऐसे मानते हैं दो इंसान है. One person is A and other person is B. दोनों ट्रेन में ट्रेवल कर रहे हैं. ड्यू टू सम रीजन ए ने बी को धक्का मार दिया बाय मिस्टेक. बाय मिस्टेक. लेकिन बी जो है सोचता है उसने पर्पसली मुझे धक्का मारा. सो व्हाट स्टार्ट्स बेसिकली अ नॉर्मल फाइट वेयर बी सेज मुझे धक्का क्यों मारा. टू ए सेज अरे गलती से लग गया. बी सेज नहीं सॉरी बोल. ए सेज नहीं सॉरी बोलूंगा गलती से लगा हुआ है. एंड सडनली देयर इज वन मोर पर्सन, लेट्स टेक सी, who comes and talks to B. देखो सॉरी भी नहीं बोल रहा है. देन बी सेज सॉरी बोल मेरे को ए को. देन ए सेज आई विल नॉट टेल. तो बी उससे झगड़ा करता है. ए भी झगड़ा करता है. सी बोलता है देखो बी को ए ने तुम्हें धक्का भी मारा और झगड़ा भी कर रहा है. मींस इट इज योर फॉल्ट. सो बी स्टार्ट्स फाइटिंग मोर. तो ए और बी के now when A and B started the fight, C came into picture. और जब C ने देखा A और B का फाइट बहुत ज्यादा बढ़ गया है, so what will happen to C, you know? C चुपचाप निकल जाता है, एग्जिट ले लेता है. अब एक्चुअल में देखो ध्यान से, A और B के बीच का फाइट C ने स्टार्ट नहीं किया. This satisfied first reaction. Enzymes accelerate the rate of reaction but do not initiate it. C has never participated in the reaction. So, and as a result, it remains unchanged. Now we need to understand one very important characteristic. So this is how we can say that the enzymes, they remain unchanged in the reaction. Let's take the third important property. Molecule of enzyme, let's understand this way. Molecule of enzyme is larger than the substrate. This is very important property. Now you might be wondering, sir, what is substrate? That will come later, but just remember this way. Molecule of enzyme is larger than the substrate molecules. This becomes again one of the very important property. Now, the point basically where the enzymes and substrate bind. So we can say the point where the substrate binds, the substrate binds to the enzyme. The point where the substrate binds to the enzyme, that point basically is called as substrate binding site. That is called as substrate binding site. But most important, what I can say, students, that this substrate binding site is nothing but it is the active site. Active site, the one that is involved in the reaction. Let's understand few more properties of enzyme. That is the fifth property. I can say that all the enzymes basically they are amphoteric in nature. Now, when I say amphoteric in nature, what exactly it means? When I say amphoteric, it means it will act as acid as well as base both. It means that the enzymes can react, enzymes can react with acid substance. Enzyme can react with acid substance as well as alkaline substance. This is what defines the enzyme as amphoteric. So as well as alkaline substance means it has the power or potential to react with both acid as well as the base. So we can say the most important point is that the enzymes most of the enzymes are proteins. Most of the enzymes are proteins, and that's why enzymes are amphoteric in nature. Let's see this physics, and the most important point is with respect to specificity. See, I give you a very simple example for time being to understand. You all know that whenever I draw a lock has a key. So every lock will open only when it gets its key. Otherwise, it is very difficult for the lock to get open. So in the same way, we need to understand that this is called as specificity. It means the lock and the key, they are specific. If it is the correct lock and key, then we can say lock will open. It means reaction will occur. But if it is incorrect, it means that the lock and key they are not matching with each other, then I can say that reaction will not occur. So what is the most important point? That all the enzymes are very much specific in their action, and for every single lock there is a single key. So this lock basically, whenever I talk about lock, I can say that this lock is nothing but it is an enzyme. So lock is nothing but enzyme, and when I talk about key is going to act as a substrate. So remember this, enzyme and substrate. So it forms basically lock and key kind of mechanism. Let's understand one more example in terms of specificity. I give you one very simple example, that is urease. Urease, no doubt it is an enzyme. So this urease enzyme will definitely act only on urea. So if there is urea, let's take for example, here I put certain compounds. Let's take this compound to be, for example, this to be sucrose, this to be urea, this to be ammonia. So these are different compounds now, for example. And here I have made one enzyme, that is urease. So urease will always go and react with urea, even though it will collide with sucrose, but it will not act on sucrose. Even though if it collides with ammonia, it will not act on ammonia. It means that the reaction will be perfect only for urea. Like for example, when you have many keys and you have to open a lock, you try the first key, lock never opens. You try the second key, lock never opens. You try the third key, lock never opens. Then you try the actual key and the lock opens. So it means urease, even though it collides with sucrose, still it will not act. It collides with ammonia, still it will not act. It will act only on urea. That is the important rule. So this is what we need to understand. The moment enzyme and substrate reacts with each other, there is a change in configuration of substrate molecule. So remember, very important point. Change in configuration of substrate molecule. Change in configuration of substrate molecules requires new enzyme. It means if I want new enzyme, then there has to be new substrate. This is again one of the very important property that we all need to focus on. Enzymes, they show a catalytic property. Catalytic property means what? Their ability to convert things. So let's understand the seventh criteria or the property. So I say it is nothing but a catalytic property. What do you mean by catalysis? It means breakdown. So every enzyme is going to show certain catalytic property. So enzymes are like inorganic catalysts. They influence the speed of biochemical reactions, but themselves they remain unchanged. Once the reaction is complete, the product is formed, enzyme retains its shape. So for any reaction, what are the things required? We required a raw product, we can say a raw material. Then this raw material has to be subjected to certain enzyme, and after subjecting it to certain enzyme, what you get is the product. And remember students, once the product is obtained, this enzyme again regains its form. So it is not going to change. That is very important point we need to understand here, that this will regain its form. So that is what is catalytic property. Every enzyme before the reaction, after the reaction remains same. And next important part, a small quantity of enzyme can catalyze larger reaction. This is very important part. So a small quantity of enzyme, a small quantity, a small quantity of enzyme can do great transformation. So when I say great transformation, what exactly it means? It can produce lots of products with that single enzyme. Now let's take for example. Let's take for example. Now this example will solve most of your doubts. For example, there is one enzyme that is called as sucrase. Now, sucrase, as the name suggests, sucrase is the enzyme that will break sucrose. So the point here is how many sucrose will be broken down by one sucrase. So we need to understand that one sucrase has the potential to break 1 lakh, or even you can say 10 lakhs of sucrose. So one sucrase can break, or you can say can hydrolyze how many? So we can say 1, 2, 3, 4, 5. 5 zeros means 1 lakh. So 1 lakh times of sucrose. So you can understand now the power of basically one sucrase enzyme. So is with respect to its own weight. So compared as compared with its own weight. So this is very important point. Okay, enzymes, they are having a great catalytic property, and enzymes are very much specific in their action that we have discussed. Enzymes, they are showing reversibility of actions. Enzyme, let's understand the next important criteria or property. That is nothing but called as enzyme optima. So when I use this word enzyme optima, what exactly it means? Enzyme optima means what is the optimum temperature or pressure that is required for the enzyme to work. So I can put it in this way. Enzymes work best in narrowly defined conditions. It means if the temperature is, let's take 10 degrees Celsius, the enzyme will work in 9 or 11, but will not change to 15. So these conditions which we are discussing now, these conditions are called optima. Very important. These conditions are called as optima. Optima means the optimum temperature. So there has to be appropriate temperature, and at the same time there has to be appropriate pH. Now we need to understand one very important thing, that the temperature is very important for enzymes to work, as well as the pH, but under certain points. Now before starting with the next part, let's understand the nomenclature, how the naming of the enzyme is done. So that becomes our next topic. It focuses on nomenclature. Nomenclature means the ability of naming the enzyme. So any enzyme, it's very easy to understand, any enzyme will always end with this word ase. Ase simply means what? Cutting. So if I put one example, if I have lactose, so if I have to break lactose, I will use the enzyme lactase. So this ase is the one that is going to break the lactose. If I am using sucrose, so if I am breaking the sucrose, the enzyme that I will be using will be sucrase. If I am having a sugar, then let's take as maltose. So maltose can be broken down by the enzyme called as maltase. So these are very important enzymes and the nomenclature. Enzymes can be named according to their functions also. So let's understand now. Nomenclature as per the functions. So there is nomenclature as per the functions. For example, for example, if I take this, there is a compound called as AH2. Now this compound has become A. It means that it has lost the hydrogen. So the compound that is involved basically in removing the hydrogen. So with this we can say now the enzyme that is involved in removing the hydrogen, it is called as dehydrogenase. This is very important way to name. And let's take one more. If there is a compound called A and I want to add carbon dioxide to it, so it becomes what? ACO2. Now carbon dioxide is added in this reaction, so the enzyme will be carboxylase. Hope you are understanding, students. If you have understood, put it in the chat box. Yes sir, we are understanding. I am waiting for your answers. Now if ACO2 is there, now ACO2 I am breaking it as A, and we are removing carbon dioxide. If carbon dioxide is lost, so we can say the enzyme will be what? Decarboxylase, no matter what is A. So dehydrogenase, decarboxylase, or let's take in this way. If there is a compound, again let's take A, and there is oxygen adding to the reaction, so it becomes AO2, that is oxidation reaction going on. The enzyme required will be oxidases. So in this way you can do the nomenclature. Now we need to understand, as per the International Code of Enzyme Nomenclature, the name of each enzyme is going to end with ase. It consists of double name. So very important what I can say, the enzyme name ends with ase. Very important. All the enzymes name will end with ase, and it is also going to have a double name. Now you might be wondering, sir, what is double name? How things are going to happen here? So remember, students, the first name indicates the nature of the substrate, and the second name basically indicates the reaction catalyzed. So double name, when I say the first name, the first name that will be, it is going to indicate what? Nature of the substrate. The first name is going to explain the nature of the substrate upon which the enzyme acts. The nature of the substrate upon which the enzyme acts. And the second name basically indicates, the reaction catalyzed. So let's understand this with an example, because examples are very much useful for you to understand the nomenclature. For example, I give you one enzyme, and let's take example to be pyruvate, or let's put it in this way, pyruvic acid decarboxylase. So when I use this word pyruvic acid decarboxylase, it simply means one thing. Now there are two names here. This is your first name and this is your second name. When I say pyruvic acid decarboxylase, it simply means what? From pyruvic, from pyruvic acid, this enzyme is going to remove, from pyruvic acid, carbon dioxide will be released. So the first name is talking about the substrate. So substrate is pyruvic acid. Second, the reaction that is getting catalyzed is the removal of carbon dioxide. Let's understand one more example that will make you better understand again in a better way. So we have the second example. The example that we are going to talk about is glutamate pyruvate transaminase. So let's put it in this way. It is glutamate pyruvate transaminase. So we have glutamate pyruvate transaminase. Now we need to understand here when I say glutamate pyruvate transaminase. So glutamate pyruvate is the first name and the second name is the transaminase. So what exactly will be happening here? From the glutamate pyruvate, there will be transfer of amino acid. So remember what is there in transaminase? Here we can say that there is transfer of amino group. So this becomes very important part. So in this way, you can do the nomenclature of the enzymes. Now coming to the most important part that we need to understand apart from this nomenclature, enzyme activity, is how the enzymes react and what is the classification of the enzyme. Because without classification, it is not possible for you to understand enzyme. So I hope all of you are ready. And if you are ready, just put it in the chat box. Yes, sir, ready. We can start with the enzyme classification because this is very important part to be taken care of. So students, if you are ready, just put in the chat box. Yes, sir, we are ready for types of enzymes. Or even I can say it is classification of enzymes. Okay, ready, ready, ready. Very good. Thank you. Let's talk about the classification of enzyme. Now this is very important. We need to understand. At the end, I will give you a shortcut to remember. So classification of enzyme. So when I say classification of enzyme, so how many classes are there? I can say that there are 6 different classes of enzymes. So let's put it in this way, students. Classification of enzyme, I can say 6 different classes of enzymes are there. And all these 6 different classes of enzymes, they play a very important, means all the enzymes are categorized only in these 6 different classes. So which is the first one? The first one is oxidoreductase. As the name suggests, oxidoreductase means what? The enzyme that is involved in oxidation as well as reduction. So we can say oxidoreductase. What is the role? I can say it catalyzes oxidation and reduction reaction. So it catalyzes oxidation and reduction reaction. You all have studied in chemistry. What do you mean by this word oxidation and what do you mean by this word reduction? So let's understand. When I say oxidation, oxidation can be addition of oxygen or even I can say it is removal of hydrogen. This is what is called as oxidation. When I say reduction, reduction simply means that it is addition of hydrogen or I can say there is removal of oxygen. So anywhere if you see this two reaction, it is purely by the help of the enzyme oxidoreductase. Now the enzyme example that I can say here is alcohol dehydrogenase. So this is very important example. So alcohol dehydrogenase is the example. How to put it in the statement form? So let's take it in this way. I will be writing one example here. So I say alcohol plus NAD plus is going to give you aldehyde plus NaDH2, means hydrogen got added in the NAD, and here the enzyme required will be alcohol dehydrogenase. It means from the alcohol group the hydrogen was removed and it was added to NAD. So this is the first class here. Now students, let's go on to the second class of enzyme. It says transferases. So when I say transferases, this becomes the second class of enzyme. Now this transferase is basically, these are the enzyme that catalyzes the transfer of certain groups. So I can say transferase basically catalyzes the transfer of, catalyzes the transfer of certain group. So if there is a transfer of group taking place, so it is going to be with the help of transferase. So transfer of certain group between two molecules. Now this becomes the most important role of second class of enzyme, that is transferases. Now here in transferases, what we have? We have example as glucokinase. Now what is glucose? Is nothing but glucose. So to glucose, what we are going to add? The phosphate group. So I put it in this way. There is a glucose. To this glucose, we are going to add ATP, that is adenosine triphosphate, and this ATP will be converted into ADP. Why? Because it becomes glucose 6-phosphate. It means on the sixth carbon of glucose, there is a phosphate attached and it becomes ADP. Now the enzyme required here will be glucokinase. You will be studying this enzyme in respiration chapter in detail about it. Let's move on to the third class of enzyme. The third class of enzyme is hydrolases. As the name suggests, hydro is related to water. Hydro is related to water. Hydrolases means what? This enzyme catalyzes hydrolytic reaction. So I put it in this way, that hydrolases catalyzes the hydrolytic reaction. It means by the help of water, we are going to break certain compounds. So it catalyzes the hydrolytic reaction. What are the examples included under the category of hydrolases? So examples that we can have is amylase as one enzyme, sucrase as one enzyme, or even it can be protease, it can be lipase. There are different examples that can be added. So I write etc. And what is the reaction here? The reaction is very simple. I take sucrose, which is a disaccharide, is broken down into glucose plus fructose. So I can say it is broken down into glucose plus fructose, and it is done by the help of enzyme called as sucrase. So this is very important enzyme here. So this is the third class of enzyme. Let's go on to the students, the fourth class. So whenever we use this word fourth class of enzyme, the fourth class is nothing but lyases. The point here is, what is the role of this lyases, what it does. So we need to understand it is involved in elimination reaction. Okay, it is involved in elimination reaction. Now when I say it is involved in elimination reaction, it simply means that it is going to remove certain compounds from here. So it is resulting in removal of group of atoms. So remember, lyases is responsible for removal of group of atoms. So if the group of atoms are removed, what will happen? There will be a double bond formed. So removal of group of atoms from substrate. Removal of group of atoms from substrate. And what happens to this if the group of atoms are removed? So we can say removal of group of atoms from substrate, leaving a double bond. So there is a double bond formed in the reaction. So let's understand this with respect to an example. So whenever we talk about lyases, the example that we have is aldolases. First example can be aldolases. Second example can be decarboxylases. That can be the second example. Then we have dehydratases. These are different examples involved. Let's understand with respect to a particular reaction. I put etc. So when I write the reaction, it says histidine. Histidine, we all know it's a type of amino acid, and it is broken down to give you histamine plus carbon dioxide. So this reaction is catalyzed by the help of the enzyme that is called as histidine decarboxylase. So when I use this word decarboxylase, it simply means what? Removal of carbon dioxide. So histidine decarboxylase becomes the enzyme here. Let's move on to the fifth class of enzyme. And the fifth class of enzyme is nothing but it is isomerases. So when I say isomerases, it is involved in isomerism. What it does, it catalyzes the structural rearrangement within a molecule. So I can say it catalyzes the structural it catalyzes the structural rearrangement within a molecule. So it catalyzes structural rearrangement within a molecule. Very important point, that the molecules, if they are arranged, it means what? The formula remains same, only structure changes. So that is what is called as isomerization. Only structure changes. Now here, the examples that we can have is epimerases. These are the examples. We have epimerases as one of the example. We have iso. We have isomerases as another example. So we have isomerases. Then we have mutases. These are different examples involved in isomerization. And I write etc. Let's put a reaction. Let's see a reaction and then we can understand what exactly it means. For example, glucose 6 phosphate. So on the glucose, on the 6 carbon, there is a phosphate group attached. So this glucose 6 phosphate is converted into fructose 6 phosphate. So glucose and fructose we all know is isomer. So glucose 6 phosphate gets converted into fructose 6 phosphate. So it is taking place purely because of the enzyme isomerase. Because we all know, students, that the formula for glucose is C6H12O6. Formula for fructose is C6H12O6. The only difference that we see is in the structure. So whenever we draw glucose structure, we draw it like this. This is what is glucose. But when I draw a fructose structure, I will draw it like this. So this is what is isomerization. There is change only in the structure, not in the formula. Let's move on to the next class of the enzyme, that is called as ligase. That is your seventh class. You can say sixth class of enzyme. Sixth class of enzyme, that is nothing but ligases. Now students, ligases, they are also called as synthetases. So remember this, ligase, basically we need to understand that it is involved in ligating, in joining. So what will be the role of this enzyme? So we can say that ligases is nothing but enzyme which catalyzes the covalent linkage. So we can say ligases are nothing but enzymes which catalyzes covalent linkage. So remember this, enzymes which catalyzes covalent linkage of the molecule. So here the two compounds will be joined to each other by the help of ligases. And remember, there is energy involved. So energy is required for this reaction to occur. So energy is utilized. Now when I say energy is utilized, we all know that the energy will be in the form of ATP. So that energy that is utilized, either it will be ATP, adenosine triphosphate, or GTP, that is guanine triphosphate. Now the point here is, what are the examples involved in ligases or synthetases? So we have glutathione synthetase. We have glutathione synthetase. So glutathione synthetase becomes one example. Pyruvate decarboxylase becomes another example. Okay, and let's take it in the form of reaction. Let's write one reaction to understand this. So it is pyruvate plus carbon dioxide plus ATP. It will be joining together to forming ADP plus PI. Now when I say PI, it is inorganic phosphate, plus it will form OAA, that is oxaloacetate. Now this will be catalyzed by the help of enzyme pyruvate carboxylase. So what we have seen here, we have seen that the carbon dioxide basically combined with pyruvate, that is pyruvic acid, and it has formed OA. If somebody asks you what is OA, just remember it is oxaloacetic acid. So this becomes the full form of OAA. Now this is how the 6 classes of enzymes are categorized. Now you might be wondering, sir, is there any shortcut to remember the name of these 6 classes? So definitely yes, students, I will give you the example. The shortcut to remember at least the names of these 6 classes. So I put it in this way. The shortcut is lethol. The shortcut here is lethol. So if I say Lithol, lethol. So when I say lethol, this can be one shortcut, or even if you want, you can use another shortcut. It says Lily teacher is hyoxy. You can use any of the shortcuts. It's all up to you. I have given you 2 shortcuts. Lily teacher is hyoxy. Now the point here is, what exactly you mean by this? When I say Lily, Li, it stands for ligase. Ly, it stands for lyase. Tr, it stands for transferase. Is basically stands for isomerase. Hy stands for hydrolase, and oxy stands for oxidoreductase. So if you remember this, Lily teacher is hyoxy, you can definitely give the answer. So if I put for lethol also, so L, let's take to be ligases, I is for isomerases, T is for transferases, H is basically for hydrolysis, O is for oxidoreductase, and we have L, that is nothing but lyases. So like this you can remember all the 6 different classes of the enzymes. Now this is the classification of enzyme, students. What we need to understand here is now mechanism of enzyme action. This is very important and very interesting topic that we need to understand, that how the enzymes will react, how they are going to form the bond, the substrate, everything. So let's take the next topic. The topic is mechanism of, the next topic is mechanism of enzyme action, how the enzymes are going to react. So the next topic is mechanism of enzyme action, how the enzymes will react. As I told you that every enzyme is going to act with the substrate. So let's put it in this way. For any enzyme to act, there has to be 2 things: enzyme plus substrate. If enzyme and substrate, they are coming together, then only the reaction is going to take place. So we have enzyme plus substrate. Enzyme I call it as E, substrate I call it as S. So it becomes what? It becomes ES complex. If somebody asks what is ES complex, you will say it is nothing but enzyme substrate complex. So this enzyme substrate complex is nothing but ES complex. Every enzyme is bound to react. We need to understand there was a scientist in the name of Koshland. Koshland, somewhere in 1959, he talked about the mechanism of enzyme reaction. And to be very much specific, he didn't focus on enzyme substrate reaction. He focused on a model that was induced fit model. But before that, let's understand this. This is very interesting and very important. Every enzyme reacts with specific substrate. It's simply like this, that every enzyme is going to fix in a particular substrate. It's like a lock and key mechanism. So even I can put it in this way: one enzyme, one substrate. So one enzyme, one substrate mechanism becomes very much important here. So it can be in the form of lock and key analogy. So when I use this word lock and key analogy, what exactly it means? When I say lock, the lock will be enzyme. When I say key, the key will be nothing but substrate. We need to understand. I draw one diagram that will help you to understand in a better way. So let us take this to be the lock. So now this lock is nothing but it is the enzyme. Okay. And I draw a different key. So this is one key. This is another key. This is another key. Okay. So there are three different keys. So based on this, what you will say? 1, 2, and 3 keys are there. So this is going to fit here. So now this is what is enzyme substrate complex, which we all call it as ES complex. So it is very important to understand that every enzyme will fit in a particular substrate, or I can say every substrate will fit in a particular enzyme. This is very important. So substrate plus enzyme, remember, it forms ES complex. Now once this ES complex is formed, what will happen? This enzyme now, this induces the enzyme to alter the shape. This induces the enzyme to alter the shape of product, basically alter the shape of substrate. Now once the shape of the substrate is altered, it will be converted into a product. Just remember this. And finally what will happen? It will form a new compound. So let's take it in this way. In a very simple way, I can explain here. This is the enzyme. Okay. This is the substrate. So enzyme and substrate, it will react together. And it is going to form what? It will be forming enzyme substrate complex. Now the enzyme will now change the shape of the substrate so that it gets converted into product. So here the shape, let's take, becomes like this. Now this is the product. So enzyme will remain as it is. There will be nothing happening to the enzyme because enzyme remains unchanged. But what is formed now? A product is formed. So now what we need to understand, student, this product which is formed is going to act as, now this product will act as substrate. This product will act as a substrate for another enzyme, not for this enzyme. So there has to be another enzyme. Let's take for example this. Now this is another enzyme, the black color one. Now for this enzyme, this product is now going to act as a substrate. Again it will react, again product will be formed, and the reaction goes on and on till it becomes the final product. So one substrate reacts with enzyme, forms the enzyme substrate complex. Enzyme changes the substrate into product. As a result, the shape of product changes, and this product will act as an enzyme. This product will act as a substrate for another enzyme. And finally, the reaction goes on. So we need to understand different models to be very much specific. Now here, if you look at this very carefully, it is actually explaining one very important model that is called as induced fit model or flexible model that was given by Coastland. So what Coastland gave, I will say Coastland gave induced fit model. Coastland gives induced fit model, or even it is also called as flexible model. Hope students you have understood this mode of reaction. And if you have understood, please put in the chat box. Yes, sir, understood. I am waiting for your answer. And after that only I will move on to this model. Yes, thank you. Yes, yes, yes. Very good. Thank you. Okay, thank you. You understood. Let's understand this now. I draw a diagram that is given in your textbook to help you to understand here. A complex flexible model. So I put in this one. Now this is one of the enzyme. This is nothing but the enzyme. Okay, here, and I draw a substrate. So let us take this to be the substrate. So what will happen here? It is enzyme plus substrate. Now this enzyme plus substrate will now combine here. So when it combines, the structure becomes somewhat like this. So this shaded portion comes here and some part goes downward. So what is this now? This is nothing but ES, enzyme substrate complex. So what will happen to this? Once the reaction is over, the enzyme will become normal as it was, but it has formed something else that was the product. So let's take it in this way. So this is what you are observing is nothing but the product. So I can say it is E plus P. Now this product will act as a substrate for another enzyme. This is one of the induced fit model. Then we have lock and key model. So remember students, lock and key is very important. Okay. There is something called as mechanism of enzyme action. In this we have lock and key model. So when I say lock and key, it means what? This model that you have studied is basically nothing but it is complex flexible model. Remember this. This is induced fit model, or this induced fit model is also known as flexible model. This is the first experiment or first mechanism of action of enzyme. So it is induced fit model, or even it is called as flexible model. The second model is lock and key model, I can say. So second model that we are going to study will be lock and key. So how it will react? Let's take different color. So I put it in this way. This is one mechanism. So let us take this. So this becomes the enzyme, and here there will be a very important part here. So let's take it in this way, this way, and we draw this. So this what you are observing here will be nothing but the substrate. So let's take it in this. This is the substrate. This place where the substrate will bind, it is called as active site. Remember, it is called as active site. So this is here enzyme and substrate. Now when they react, what will you get? So let's make it as a shaded portion so as to distinguish the different enzyme and substrate. So here again, the story is going to start like this, in this way. So I put it here this way, and I will draw the substrate. So the substrate is now going to fit into this. So remember this. The substrate is going to fit into this. So this becomes what? This becomes enzyme substrate complex. It is fitting properly, and finally what happens? This enzyme will get separated and there will be formation of product. This enzyme gets separated and there will be formation of product. So what are the two products that will be formed? So let us take this and this. These are the products. So look at the product. The shape of the product has changed. So this product now will act as a substrate for another enzyme. So this kind of model where you can see proper fitting, it is called as what? Lock and key model. This is another one. So here the enzyme and the substrate complex is formed. So this becomes your enzyme and this becomes the product. So I can put it in this way, students, just to make you things easy, clearly understand. Let's take this enzyme to be sucrase and the substrate to be sucrose. So when sucrase combines with sucrose, it gives you a product. So now the product will be what? The sucrose will be broken down into glucose plus fructose. This is very important part. So this is what we need to understand. This is lock and key mechanism where one correct size key, that is a substrate, fits into the keyhole active site of the lock, that is nothing but the enzyme. So remember students, the binding of the substrate is very important. And when the substrate binds to the enzyme, it can cause change in the distribution of electrons in the chemical bonds of the substrate and ultimately causes the reaction that leads to the formation of product. So every enzyme has a fixed substrate. That is what we need to understand here. This was basically the enzyme substrate reaction. I hope you have understood. And if you have understood this enzyme substrate reaction, just put in the chat box. Yes, sir, it's clear. So there are different enzymes, there are different products. So if it is clear, just put in the chat box. Yes, sir, clear. So that we can move on to the next important part, because every now what we are going to see, we are going to see the factors affecting enzyme. So if you have understood, just let me know. Yes, sir, it is clear. We can start with the factors affecting enzyme activity. So if you have understood, just put in the chat box. Okay, clear, clear, clear. Let's take it in this way now. Very good. So we are starting with a new topic and the most important topic, that is factors affecting the enzyme activity. So we have factors affecting the enzyme activity. Factors affecting the enzyme. There are different factors that will affect the enzyme activity, and we are going to study each factor in detail. And during this explanation, there will be different graphs also coming into picture. So let's understand the very first factor. The very first factor will be temperature. We all know that the enzymes basically they are proteins. We all know that enzymes, they are nothing but proteins, and we all know that the proteins basically at high temperature, proteins at high temperature gets denatured. Very important. And you can say proteins basically at low temperature becomes inactive. So whether temperature is high or temperature is low, the enzyme activity is going to decrease. The point here is, what is the optimum temperature for the enzyme activity to work? So when I say temperature, there is a very important word that is called as optimum temperature. So let's put it in this way, students. When I say optimum temperature means what? Where the enzyme activity will be maximum. So optimum temperature is the body temperature. What is the body temperature of humans? So we all know it is 37 degrees Celsius. So all the enzymes will be at its maximum at this optimum temperature, because we all know that humans are homeotherms, and we have studied this in kingdom Animalia, that the mammals, they are homeotherms. So the rate of chemical reaction increases. Even at the temperature above 40 degrees Celsius, things will change. So let's put it in this way. I take it as 37 degrees Celsius. If I increase the temperature, okay, so let's take the temperature goes till 40 degrees Celsius. Till 40 degrees Celsius, what I will say, the enzyme activity will decrease. But beyond 40 degrees Celsius, the enzyme gets denatured. So we need to understand, beyond 40 degrees Celsius, the enzyme gets denatured. And if the temperature is decreased, so let us take to becomes at 25 degrees Celsius, or let's decrease to be further this. So what will happen? I will say that the enzyme becomes inactive. See, when I say denatured, it means it is totally lost. It cannot be regained. But if the enzyme is inactive, it means it can be regained back. So remember students, when I draw a graph with respect to this, you will understand here that how the graph works. So let me draw this enzyme activity graph. So I put in this way. This is the x-axis and the y-axis. So what I will be doing on the x-axis, I will be writing, let's take temperature in 0, let's take in degree Celsius basically. And on the y-axis, we are going to put enzyme activity. So we can put it as increasing enzyme activity. So what we need to understand here, when the temperature, let's take to be here 0, and let's take to be here this point to be 37, and this point to be 40, and here let's take to be 60. So as the temperature is at 0, you know, so what will happen, the temperature enzyme activity will be less, but as it moves towards 37, you see it is increasing, and when it crosses 40, the temperature, or you can say increase in temperature, will decrease the enzyme activity. It means what we see, that somewhere around at 40 degrees Celsius, the enzyme activity is maximum. So this point becomes what? This point becomes optimum temperature. And if I go towards the left side of the optimum temperature, enzyme activity decreases. If I go towards right side of the optimum temperature, enzyme activity decreases. Optimum temperature means what? Optimum temperature means that the temperature at which the enzyme activity, the temperature at which the enzyme activity is maximum. So what you need to understand here, that at high temperature the enzyme gets denatured, at low temperature the enzyme is inactive. So the optimum temperature for enzyme to work will be around or between, you can say, 37 to 40 degree Celsius. This becomes the first factor affecting enzyme activity. The second factor that we need to focus on is the pH. Now you all have studied in chemistry what is pH. So I put it in this way, pH is nothing but it is negative logarithm to the base 10 concentration of H plus ion. So this is what is pH. You all have studied the pH scale also. The pH scale is going to range from 0 to 14, and there is a 7. So here what I will say, from 0 to 7 it is acidic condition, from 7 to 14 it is a basic condition, and exactly at 7 what is there, we say it is a neutral condition. So what we need to understand here, whenever it comes to pH, all the enzymes basically they will be more active at a basic pH as well as the acidic pH depending on the nature of the enzyme. So what we need to understand here, let's put it in this way with respect to graph, that will be very easy. So I take this 0, then I take this as 7, and I take this as 14. If the pH is shifting from 7 to 14, the EA means enzyme activity will keep on decreasing. And if I take it from 14 to 7, then the enzyme activity will increase. If I go from 0 to 7, the enzyme activity will increase. And if I go from 7 to 0, enzyme activity is going to decrease. So with this, what you understood? You understood one thing, that the enzyme, you understood one thing, that the enzyme activity is maximum at which pH? We can say neutral pH. So remember, all the enzymes, they will be active maximum at the neutral pH. But some of them can be acid as well as base also. So if I have to draw a graph now, let's understand the pH with respect to graph. So here on one side I put it as pH, So let's take 0, 7, 14 on the x-axis. On the y-axis, I will put as increasing enzyme activity. On y-axis, I will put it as increasing enzyme activity. So when I draw a graph, what I will see? I will see from here, see it is increasing, 7 it is increasing, and after that it has decreased. So what is that optimum temperature here? Optimum pH, basically not the temperature, is somewhere around, I can say, 7. So this becomes your optimum pH, the pH at which the enzyme activity will be maximum. So remember students, as far as pH is concerned, all the enzymes will be best active at the pH that is neutral. Moving towards the third and the most important factors affecting the enzyme activity is the substrate concentration. And if you understand this, then things are very easy. It is substrate concentration. So now what is this substrate concentration? It depends on the number of enzymes and the substrate. See, I put it in this way that the rate of reaction of enzyme, rate of reaction of enzyme increases with increase in substrate. If I increase the substrate, no doubt enzyme activity is going to increase. But beyond certain concentration limit, because there is always a limit to all the reaction. So beyond certain concentration, what will happen? Increase in substrate, increase in substrate concentration will not result in increase in rate of reaction. Now to understand this, I have to give you a very simple example, and this example will guide you well to understand it. Let us take that there are 100 chairs. There are 100 chairs. And there are, let's take 200 men. So logically what happens, these chairs, basically they are going to act as an enzyme. And these men, they are going to act as a substrate. Now, these 100 chairs, they are basically present in a room. They are present in a room. So let us take these all to be chairs. There are 100 chairs present in a room. Now the men, they are waiting outside the room. So this men is nothing but they are the substrate. So as the men enters in the room, they will start to sit on the chair. They will occupy all the chair. But if when all 100 chairs are occupied, it means all the substrates they have bounded to the chair or the enzyme. Now if I increase the substrate, if I keep on increasing the men, will there be any seat available? No. And it means what? Can they sit on the chair? No. So will there be any reaction? I can say no. It means if I increase the enzyme, no doubt as the substrate increases, the enzyme reaction increases, but up to limit. After that, there will be no such reaction taking place. Let's understand in this way now. This concentration of substrate is very important. There is always a limiting level. So what we need to understand, there is something called as Vmax. What is Vmax? Vmax is nothing but it is the maximum velocity that can be achieved in any of the reaction. So let's understand this. Now for this, I need to draw first of all a graph for you, and then after that we can discuss what is Vmax and everything. So I take this here. Okay, so let us take this. Now to this we put it in this way. This is what is x-0. This I take it as x-axis. This I take it as y-axis. So on the y-axis there is something called as velocity, the velocity of reaction, or the velocity at which the enzyme substrate bind to each other. And here what we have is nothing but the substrate concentration. So on the x-axis we are having the substrate concentration. So as you increase the substrate concentration, the rate of reaction will increase. No doubt the velocity of the reaction will increase, but after a certain point of time it is going to become constant. So let's divide this in three different range. So here, this is the maximum peak, where the reaction is achieved. So this point I can call it as V max. What is V max? The maximum velocity. So I put it as V max. The maximum velocity that can be achieved in any of the reaction, and I put this as C. And this is V max. Somewhere here will be the half V max. Half V max means the velocity achieved basically with respect to half time required. So half V max I put it here. This becomes my half V max. So the velocity at which half of the reaction will be covered, basically I put it this way. So now this, wherever you get a drop of this here, this is called as Km. Very important. So this is A, this is B, and this is C. So half Vmax is here. What is Km? Km now is very important concept that we need to understand. Km is equal to Michaelis Menten's constant. What is Km? We say it is Michaelis Menten constant. It means Vmax is, let's take it in this way. If the Vmax is achieved at number 10, this is just an example to understand. If Vmax is achieved at number 10, then I can expect that my room will be half filled by what time? So I can say 5. So that 5 I take it as Vmax by 2, and I drop it, and finally I get a Michaelis Menten's constant. Okay, so it is half Vmax. This is concentration of substrate. Three distinct phase: A phase, B phase, and the C phase. So Km is nothing but it is the Michaelis Menten's constant. It is defined as substrate concentration. What is Km? So we can say that it is defined as substrate concentration. It is defined as substrate concentration. Now this substrate concentration will be expressed as what? We can say it is expressed as moles per liter. So Km is Michaelis Menten's constant defined as substrate concentration to produce half of maximum velocity in an enzyme. To produce half of maximum velocity, in an enzyme catalyzed reaction. This is very important to understand. Half maximum what will be achieved. So we need to understand this is 50%. So here at Vmax, what I can say, at Vmax I can say that 50% of chair is occupied. Right now, this is 50% of chair is occupied. That is what is called as Km, or half Vmax. If I take this example of 100 chairs and 200 men. Km value is a constant feature for any enzyme. Just remember. So let's understand the value of Km. So Km, whenever we talk about the Michaelis Menten constant, we need to understand some important point with respect to K. First, what I can say, Km is a constant. It is not going to change. It is a constant characteristic feature of an enzyme. It is a constant characteristic feature of an enzyme. So for particular enzyme, the Km is going to remain fixed. It means every different enzyme is going to have a different Km value. What exactly it is representing? So I can say Km, it is representing, the Km is representing, the Km is representing the Michaelis Menten constant. Basically, it is representing the strength of ES complex. It is representing what, measuring of strength of ES complex. So remember this, measuring the strength of ES complex. How much strong is the enzyme substrate complex. It can be measured by the value of Km, that is Michaelis Menten constant. Let's understand it this way. There are now different conditions. I take Km, that is Michaelis Menten constant. First condition is that there is a low Km value. The Michaelis Menten constant that you have obtained is low. And if it is low value, it indicates what? Strong affinity between enzyme and substrate. If there is low Km value, what it indicates? Strong affinity. If there is low Km value, it indicates what? Strong affinity between enzyme and substrate. So it is enzyme and substrate. So it is strong affinity between enzyme and substrate. But if there is high Km value, if the Km value is very high Km value, the Michaelis Menten constant value is very high. So under this condition, what exactly it is indicating? It reflects a weak affinity between enzyme and substrate. So this high Km value is going to reflect what? Weak affinity between enzyme and substrate. So we need to understand here the Km value has to be low if you want the reaction to take place at a faster rate. High Km is going to indicate weak affinity. For majority of the enzymes, now this is very important. I can put it in this way. For majority of enzymes, the Km value, the Michaelis Menten constant value, will be in the range of 10 raised to 5 to 10 raised to minus 2 moles. So 10 raised to minus 5 to 10 raised to minus 2 moles is the range for the Km value. So this is what is Michaelis Menten constant, one of the factor, basically concentration of substrate. Now going to the next important point. We have talked about substrate concentration. Now let's talk about the next one, that is the fourth one, which says enzyme concentration. This is again one of the important factor affecting the enzyme activity. It is enzyme concentration, because the chair also has to be in fixed number. If I keep on increasing the chair, then definitely substrate increases, no issues as such. So let's put it in this way. If I have to put it in a simple form, I can say that rate of an enzymatic reaction, rate of an enzymatic reaction is directly proportional to the concentration of the substrate. Is directly proportional to the concentration of the substrate. More the concentration of substrate, more will be the enzymatic reaction. So even I can say one more point here, that the rate of reaction, is also directly proportional to square root of the concentration of enzyme, is also directly proportional to square root of the concentration of enzyme. So these are the two important statements that we need to understand. First, rate of enzymatic reaction is directly proportional to concentration of substrate. Second, rate of reaction is also directly proportional to the square root of the concentration of enzyme. It means what? It means as as the enzyme concentration increases, the rate of reaction increases. This is very important statement. Now let's understand this with a particular graph, because graph will help you to understand in a better way. So again, I draw a simple graph for you so that you can understand things very clearly. So I draw this and I draw some lines for you that will help you to understand. Then we will write x-axis, y-axis and all. So let's take this to be the one line. Now let's take another line. These lines are very important because they are indicating the concentration of the enzyme. Okay, so based on this, so I put in this way. So I take this all. Now let's understand how the things are working here. So again, here I will take x-axis. Okay, let's take one more line. Basically, that line will help you to understand again in a better way. So from here, I take this, I take it here. So let's take this to be the x-axis, this to be the y-axis, and this to be 0. On x-axis, we are having time, the time taken. On y-axis, we have the product concentration. So I put it as product concentration. Now this red line is basically with no enzyme. If there is no enzyme, you can see that time taken is very high, but the product is very low. This is with 1x enzyme. If I use 1x concentration of enzyme, the product concentration is very fast and the time taken is less. Here if I take 2x, then again the things increases. Then if I take here 4x, the reaction is very fast. So what we understand with the graph, as the enzyme concentration increases, the rate of reaction also increases. So this becomes a very important point. So I write it here. As the enzyme concentration increases, the rate of reaction increases, and definitely the time taken will be less. So this is the another factor affecting the enzyme concentration. Other substances that will affect the enzyme concentrations are, so that will be the next factor, that is factor number 5 affecting the enzyme concentration. I put it as other substances. So other substances that is affecting the enzyme concentration. So what we have, the enzyme action increases, like you can say cofactor. Cofactor is another substance. Then we have coenzymes as another substance. Then we have activators as other substance. Then we have inhibitors as other substance. So all these substances basically, if they increases, what will happen? Rate of reaction will change. Now the point here is it going to change always in increase? No. So we can say decrease or increase will result in the change in the rate of reactions. Let's understand inhibitors, they are going to play a very important role here. So the next part that is very important to focus on basically is inhibitors. And case of inhibitor, students, you need to understand there are two types of inhibitors that will play an important role. The first type of inhibitor is the competitive inhibitor. Competitive means the inhibitor and the substrate basically they both compete to bind at the active site of enzyme. And when we talk about non-competitive inhibitor, it simply means one thing, that the inhibitor and the substrate they will not compete. In fact, the inhibitor will bind to some other part of the enzyme and will change the nature of enzyme. So here I can say in case of competitive inhibitor, the enzyme nature is not changing. The enzyme nature is not changing, it is going to remain constant. But here in case of non-competitive inhibitor, I can say that the enzyme nature totally changes. It totally changes and everything is new. So in case of competitive inhibitor, the example that I can take is bacterial pathogen. When you have bacterial infection, that time there is a competitive inhibitor working. But in case of cyanide poisoning, but in case of cyanide poisoning, I can say that is non-competitive inhibitor. So this is what we need to understand here. This is competitive and non-competitive inhibitors. Few very small but very important definitions, students, that we all need to know, because in your exam they can ask what are the meanings of these terms. So let's understand this. The very first term is, I put it as apoenzyme. If somebody asks you now what is apoenzyme, you will say apoenzyme is nothing but protein component of enzyme. So the protein component of enzyme, the component of protein component of enzyme is known as apoenzyme. Next word that we need to understand is a cofactor. What is cofactor? Non-protein component of an enzyme. So non-protein component of an enzyme. So non-protein component of enzyme is known as cofactor. Then we have something called as holoenzyme. So when I say holoenzyme, basically it is apoenzyme plus the cofactor. So apoenzyme plus the cofactor is known as holoenzyme. Cofactor, if you remember, we have studied, it is also known as prosthetic group. So this prosthetic group becomes the holoenzyme. Then you might come across a word that is called as isoenzyme. So what do you mean by this word isoenzyme? Remember students, isoenzyme means what? Different molecular structure but same function. Different molecular structure but same function. Different molecular structure but same function is known as isoenzyme. Then we have something called as inducible enzyme, but that inducible enzyme is not there for you, so I'm not going to touch that part. This is what we need to understand, the factors affecting enzyme and the enzymatic activities. Let's understand one very important concept. It is not there in your textbook, but it is very important as far as NEET exam is concerned. So NEET exam, if you are appearing for NEET exam, then it will help you to understand here. That is activation energy. It talks about concept of activation energy. For any enzyme to act, concept of activation energy. For any enzyme to work, it requires some energy. And if there is no energy available, then enzyme reaction will not take place. So for that, let us draw one graph to understand this activation energy. So on this, let us take it in this way. On x-axis, we are going to take rate of reaction. On x-axis, let us take it as rate of reaction. On the y-axis, here we will take it as potential energy. We will take it as potential energy. And let us take this. Now, this dot that I'm drawing, this line is very important. This line is nothing but, it is the optimum, basically, half. So let's understand this. If I draw the first here, so I start from here, I draw this and I bring it back here. This is the first graph. And let's take at the second graph. So here I take the second graph where it is going to raise, and again finally it will be converted into. So what we need to understand here, students, this is nothing but the substrate. This is nothing but the substrate that we all call it as S. This is activation energy. This line that you observe from the basic point is the activation energy. So here I can say, here this is nothing but the raw material, and this side what we have is the product. This is raw material and this is nothing but the product. So here, this is activation energy with enzyme. If there is enzyme present, so activation energy required is very less. So I can put it in this way: activation energy with enzyme. So if the enzyme is present, the activation energy is less. But if enzyme is not present, look at the activation energy, it is very high. So we can say this is activation energy without enzyme. So this you will be studying in chemistry basically, of the activation energy. So what exactly happens here? What we need to understand here? The important point is activation energy, actually what it does. So I can say, students, that the activation energy, it increases the kinetic energy of the system. Activation energy increases the kinetic energy of the system and brings about forceful collision between the reaction. This is what we need to understand. Brings about forceful collision between the reaction. So this is all what you should know about the enzymes. And I hope you have understood the concepts clearly. And if you have any doubt, you can put it in the chat box. And if it is clear, students, just put in the chat box, yes, sir, clear. I'm waiting for your answers.